In this review, we discuss tumor clonality and resistance mechanisms in EGFR mutation-positive non-small cell lung cancer and the implications for therapeutic sequencing of EGFR TKIs. 3 generations of EGFR TKIs are available for first-line treatment of EGFR mutation-positive NSCLC. The first generation, reversible EGFR TKIs, gefitinib and erlotinib. The second generation, ERB-B family blockers, afatinib and dacomitinib. And the third generation, irreversible wild-type sparing TKI, osimertinib. Recent head-to-head clinical trials have demonstrated that afatinib, dacomitinib, and osimertinib are superior to first-generation TKIs in a first-line setting. Regardless of which EGFR TKI is chosen, most patients will become resistant to therapy. First- and second-generation EGFR TKIs have similar resistance mechanisms. The most common mechanism is the emergence of the so-called gatekeeper T790M mutation in exon 20 of EGFR. Fortunately, second-line osimertinib is an effective treatment option in this setting. T790M independent mechanisms of resistance are less well understood. Osimertinib is also indicated for the first-line treatment of EGFR mutation-positive NSCLC. However, mechanisms of resistance appear to be highly heterogeneous. Consequently, targeted treatment options following osimertinib failure remain uncertain at this time. Ultimately, optimal treatment strategies in individual patients with EGFR mutation-positive NSCLC will be facilitated by analysis of how the tumor evolves over time. Tumors evolve over the course of their development and during therapy. Genomic instability of developing tumors creates a genetically diverse cell population that is subject to selective forces. Early oncogenic mutations that drive tumor development, such as EGFR mutations, tend to be present in all tumor cells and regions. In contrast, branch mutations are later subclonal events that appear in a small number of tumor cells and regions. Any of these cells that are resistant to therapy could proliferate into a resistant tumor. Therefore, in terms of treatment of EGFR mutation-positive tumors, it would be highly advantageous to identify subclonal background mutations and monitor them longitudinally over the course of treatment. It is becoming increasingly clear that EGFR mutation-positive tumors are genetically heterogeneous. Recent evidence indicates that in some tumors, small numbers of T790M positive cells pre-exist prior to treatment initiation. On treatment with first or second generation EGFR TKIs, these cells are subject to strong selective pressure and develop into a resistant tumor that can be subsequently treated with osimertinib. However, not all tumors have pre-existing T790M positive cells. In some cases, T790M arises over the course of treatment and is consequently only present in some cells of the resistant tumor. Of course, as mentioned, some resistant tumors have no T790M positive cells at all. Technological developments have identified sources of heterogeneity in EGFR mutation-positive tumors. One source of heterogeneity is the existence of uncommon EGFR mutations. Some tumors harbor more than one type of EGFR mutation, and these can exist on the same allele, so-called compound mutations. Some uncommon mutations, particularly compound mutations, are known to be insensitive to EGFR TKIs and can therefore drive the development of resistant tumors. Another source of heterogeneity is the existence of genetic aberrations that coexist with EGFR mutations. Recent data have indicated that additional mutations are very common in late-stage tumors and include known oncogenic drivers. Additional mutations are often present within T790M positive tumors that are resistant to first or second generation EGFR TKIs. These observations have implications for the choice of treatment. Ideally, TKIs should have a broad inhibitory profile to mitigate for the heterogeneity of tumors and the risk of early expansion of resistant subclones. Also, if T790M-mediated acquired resistance occurs, it would be advantageous that the T790M clone is as homogeneous as possible to maximize the chances of a prolonged response to osimertinib. Emerging data indicate that different TKIs could have a different impact on how tumors evolve. For example, we have undertaken experiments that have specifically assessed the T790M allele frequency of tumor cells following acquired resistance. We cultured EGFR mutation-positive cells, subjected them to mutagenesis, and then cultured them in the presence of afatinib or erlotinib for 3 weeks. As expected, the majority of resistant clones were T790M positive. Interestingly, the frequency of T790M alleles was higher in afatinib-treated cells than erlotinib-treated cells, indicating that T790M clones might be more homogeneous following afatinib treatment. Preclinical evidence indicates that second-generation TKIs may have a broader inhibitory profile against uncommon EGFR mutations than first or third-generation TKIs. This figure illustrates the cellular in vitro activity of gefitinib, erlotinib, afatinib, and osimertinib against over 100 uncommon EGFR mutations, including compound mutations. Blue indicates sensitivity. As can be seen, afatinib appears to have broad activity, particularly against compound mutations. While few clinical studies have investigated the activity of EGFR TKIs against uncommon mutations, afatinib has demonstrated activity against certain uncommon mutations in clinical trials, including L861Q, G719X, and S768I, and is indicated for use in this setting. We speculate that the superiority of second and third generation EGFR TKIs could potentially be explained from a clonal evolution perspective. For example, the impressive clinical benefits with osimertinib in the FLAURA trial probably reflected strong inhibitory activity against T790M, thus preventing the clonal expansion of pre-existing T790M cells or the development of de novo T790M positive clones. Furthermore, as illustrated in the figure, the broader inhibitory profile of second-generation TKIs against other ERB-B family receptors or particular mutants could prevent emergence of tumor cells that would otherwise survive treatment with first-generation TKIs. This, in turn, could delay the onset of clonal expansion and acquired resistance to therapy, which is possibly reflected in the observed improvements in PFS with afatinib and dacomitinib versus the first-generation TKI gefitinib in the LUX-Lung 7 and Archer 1050 studies, respectively. This hypothesis could also explain recent observations of encouraging clinical benefit with sequential second and third generation EGFR TKIs. The recent observational GEOTARG study assessed outcomes in 204 patients who received sequential osimertinib after first-line afatinib. Overall median time on treatment was 27.6 months and was particularly promising in Asian patients, 46.7 months, and patients with an exon 19 deletion mutation, 30.3 months. In a post-hoc analysis of LUX-Lung 3, 6, and 7, median OS was not reached in 37 patients who received sequential afatinib and osimertinib. The 3-year survival rate was around 90%. In summary, this article demonstrates that clonal evolution plays a key role in acquired resistance of tumors to treatment. EGFR mutation-positive NSCLC tumors are heterogeneous and undergo clonal evolution. Different EGFR TKIs are likely to confer different selective pressures on developing EGFR mutated tumors. In the future, further technological developments and the monitoring of tumor evolution during treatment is likely to be instrumental in guiding the optimal treatment strategy in patients with EGFR mutation-positive NSCLC.